give our remarks. Whenever you're ready. All right. Hi everyone, thank you so much for coming and joining me today. For my honors project, I'll be looking into urban development as an indicator of harmful algae blooms across Austin, Texas. So many of you probably have heard about harmful algae, especially in 2019 when there were cases of dogs accidentally ingesting fresh water that was contaminated with cytotoxins and cyanobacteria and harmful algae blooms. And then not only that, but locally the Texas Tribune article also wrote about harmful algae and how it can be a problem for not only people, or not only dogs, but also people in the article. So for what harmful algae blooms are, they're overgrown by diverse microorganisms, including phytoplankton and cyanobacteria. Of the three cytotoxins that are present in cyanobacteria is microcystine, slenderopsin, and antitoxin A. This is a table provided by the EPA listing these cytotoxins, but as well as the different species within cyanobacteria that also produce cytotoxins. So in the list, you'll notice that some of the names are repeated, so it'll show that not only do they produce one cytotoxin, they also produce multiple. In this list, you'll see that microcystine has the most known variants, and subsequently, it's also the most common in the United States. As I mentioned beforehand, these cytotoxins are harmful for humans, but also for animals. So in the list here for the health effects, as well as the organs, they impact the liver and the nervous system and other health symptoms as well. So now that we know that harmful algae blooms are a problem, what they are, next I'm going to talk about what factors contribute to harmful algae blooms. The research has shown that sunlight and warmer water temperatures and excess nutrients such as phosphorus and nitrogen creates this ideal environment where cyanobacteria are able to bloom. It's important to know that cyanobacteria is naturally occurring in our water, so any freshwater or saltwater. But what makes it harmful is when these cyanobacteria are very large quantities and able to produce more cyanotoxins that way. And when cyanobacteria create these large blooms, most cases it's because of us changing the conditions, the environmental conditions, and causing them to grow even more. So something like urban runoff or agricultural runoff, adding nutrient loadings into our creeks. Other studies have shown that nutrients is a limiting factor in algae blooms, so adding any extra is almost like a fertilizer helping them to grow. So even though cyanobacteria is naturally occurring, we're adding to the damage that they're doing by making them more frequent and more intense. So that leads to my research question, which is what are the indicators that can help or that can predict harmful algae blooms? The goal of my study is to look into what predictors within an urban and suburban setting to see what indicators can help predict harmful algae blooms as well as the presence of cyanotoxins. So I sampled 6 different creeks, 3 of which are urban, so they're located on the center of Austin here with the blue circle, and then 3 suburban creeks located here with the red circle, which is farther away from the center. And within these creeks, they were interesting, or we selected these creeks because they were most common recreational places, or they were previously reported with harmful algae blooms. And initially, these creeks were categorized by either urban or suburban by estimated visually of impervious cover. So when I saw that there were a lot of impervious cover categorized by urban, or low impervious cover categorized by suburban. But the goal of my study also looked at the percentage of impervious cover and seeing if we could use that as a proxy for development. So for the methods of measuring harmful algae bloom indicators, I looked at impervious cover. This data was provided by the city of Austin and the year 2020, and I used that data to determine if we could use percent of impervious cover as a proxy for urban development or the creek types. water temperature using a hydrotech multi-probe. And then I also measured a portion of nitrogen and phosphorus, which is nitrates and orthophosphorus, with the hatch method in the lab. Fourth, I measured the water toxicity, so I looked into microcystine, cylinder opsin, and antitoxin A. These samples were collected and sent to Austin laboratories to help measure those. And then lastly, I ran a general linear index model, looking at using the R program, and for the dependent variable is the 3 toxins that I mentioned, and the independent variable is the 8 different indicators that I measured. And since my sample size is relatively small, we use a significance of p less than 0.1. So for my results, impervious cover as a proxy of development. As you can see on the x-axis is the different things that I categorized, so either urban or suburban, and then the y-axis is the percent impervious cover. In the blue box there is the urban, and the red is the suburban. So you'll notice that there's a significant difference in the percent of impervious cover, and this is acceptable to use percent impervious cover as a proxy for development. Next, all cyanotoxins differ significantly from the percent of impervious cover, so for A, B, and C. This one is microcystine, cylinder opsin, and antitoxin A. And all x-axis of each graph is the percent of impervious cover, with the y-axis being the concentration of each toxin. So I found that as impervious cover increased, there were also increases in the toxin of each of the ones that I measured. And with impervious cover, it collects rainwater because the rainwater isn't able to filtrate into the ground. So when rainwater isn't able to filtrate, it collects any chemicals, any animal waste, any fertilizer, and it ends up in our streams, causing a major flooding. That might be a reason why we saw some more concentrations as impervious cover increased. Furthermore, we looked at water quality parameters, and they were different between cyanotoxins. For example, for microcystine, we saw that higher levels of conductivity led to or found increased levels of concentrations of microcystine. And then for cylinder opsin, we found that higher levels of dissolved oxygen related to the concentration of cylinder opsin. And for conductivity, same thing for impervious cover. If any dissolved solids or chemicals can alter the water, increasing the levels of conductivity. And for dissolved oxygen, in general, cyanobacteria is a photosynthetic bacteria, which produces oxygen as a byproduct. So dissolved oxygen may be a result of, but not an indicator per se for the concentration of cylinder opsin. In conclusion, I found that impervious cover, conductivity, and dissolved oxygen may be useful indicators of harmful algae blooms and the presence of toxins. And for future research, it would be interesting to see if these patterns hold true for a much larger sample besides Austin, Texas. Harmful algae blooms not only affect Austin, but also worldwide. And as well as looking at different seasons, I measured this during the February and March months, but it's known that harmful algae blooms are more active during the summer season, so it'll be interesting to look into that. And for acknowledgements, I wouldn't be able to complete my honors project without all these lovely individuals here. My mentor, Dr. Croquet, as well as Dr. McCracken, and then the City of Austin Watershed Department. So Brent, he provided some materials for me to be able to measure some water quality parameters, and then also guided me knowing which streams are more popular for the public, as well as during the watershed department, they didn't look at creeks, but they mainly looked at lakes and something like that, so added data for them. And then Austin Water, Angel and Crystal helped me measure the cyanotoxins, because it's very expensive to measure on my own, so they're very helpful in measuring that for me. And then the St. Edward's department, so Dr. Watley and Zoe, who allowed me to use the labs during measuring concentrations. Thank you.
Any questions for Alice? topic. And so I might have missed it, but so beyond just doing more research, which is clearly needed, the article you put up at the very beginning, it said something like there's nothing we can do about these harmful actions. Is that true? Is that?
I think it is kind of just an article saying like, oh, it's not something to be distressed about, but I think there should be something to look into. Just there's not enough research about it at the moment.
Okay. One tiny unimportant question. I really like your graphs. What program did you use to make this?
Oh, I use R.
R. That makes sense.
Thank you.
I have a question.
Yeah.
So when algae make the cyanotoxins, correct?
Yes.
And they clearly are making them to try to kill dogs. So why do they?
It's believed that they use the cyanotoxins in the water to fight off competitors. So they produce that toxin as a way to be the main organism in that water taking the nutrients.
Yes, so as far as mitigation goes, is there any evidence or research looking at using like the microbial community found in the water to somehow increase competition and outcompete cyanotoxins or cyanobacteria? Or would the mitigation mainly be more towards removal of runoff or like mitigation of runoff problems?
When I was reading, I think I've seen more mitigation efforts. So they explain how instead of adding solid impervious cover like roads, we can add maybe gravel, something to allow that filtration. That's something I saw recently.
Any other questions? We can thank Alice again.
So, I first saw Alice give a talk at I-4, I believe, last year. I was like, she's good. She knows what she's doing. So she approached me about doing a project, and I said, absolutely, let's do something. And she followed through a couple of examples, like some of those graphs we saw, maybe first tell us most of that was her. Like, she took that and she ran it, and I was really impressed. The other piece I really liked about Alice was that she wanted to do this project, and I knew we needed to engage the city of Austin. I knew that she needed resources beyond what I could provide her, but I could submit it. And she took it, she ran with it. She made phone calls, she made emails, she connected with folks, she met them in the field. It was just that kind of singular project that was really impressive. It was very good. So I will